Good morning, good morning, students. As we have studied in the previous part of this chapter, there are the different methods or the contraceptive methods to prevent the pregnancies. But there is around 2% population in the world which are not able to reproduce, or you can say they are infertile. In such cases, the role of the reproductive health is to assist them, to give them the information that how this problem can be overcome. So if the individual is not able to produce the young ones in spite of unprotected means without the use of contraceptive method for at least a 2 year in continuation, then the individual is said to be infertile. So the infertility reason may be due to the physical disorder, any type of their congenital, the disorder due to the birth, due to the drugs, or even the infections. In the just we have studied that in the reproductive tract infection may be the immunological reaction where the immune system is responding very fast and destroying the gametes or the germ cells, and the psychological thought of an individual. These can be there. And remember, out of these 2%, 70 to 80% are those which are suffering from minor disorders or the infections, which can be cured very easily by a simple providing them a simple treatment. But those other than those, there should be a specific method if the genitalia is not properly functioning, or if the uterus or the vagina is not properly developed. So in such cases, some of the corrective methods or the techniques have to be used. These corrective or techniques are known as assisted reproductive technology, or in short form they are known as arts. These arts include, number one, very common art is in vitro fertilization, or also known as test tube babies. In this program, the ovum and the sperms, they are fertilized outside the body of an individual females. That is why it is called in vitro fertilization, where ova is taken from the donor female and the sperms from the husband are collected, and when the zygote is formed in the laboratory under the similar condition as in the body, such conditions are called as in vitro fertilization. And in this case, the zygote or the early embryo is then transferred to the fallopian tube or the uterus of the female from where the ova has been taken. So this method is also commonly known as embryo transfer technique. Now this embryo transfer can be of further two types. I again repeat, in vitro means fertilization is taking place outside the body under the controlled condition and forming an embryo. Now this transfer is of two types. Number one is called as a ZIFT, zygote intrafallopian transfer. As the name suggests, when the zygote is in the 8 blastomere, they are transferred into the fallopian tube. That is, 8 cells have been formed after the zygote that is transferred into the fallopian tube. So that condition is called as a ZIFT. Otherwise, when the embryo more than the 8 blastomere stage is there, they are directly transferred into the uterus cavity. So that technique is known as intrauterine transfer. So both together are known as embryo transfer method, and they are in vitro method because the fertilization has taken place outside the body cavity, but the embryo or the zygote has been transferred either in the uterine cavity or in the fallopian tube. The next method for assisted reproductive technology is known as GIFT, gamete intrafallopian transfer. This is done in the case where the female is unable to produce the ova. So the ovum is collected from another donor female and they are introduced into the fallopian tube of the female who is unable to produce the ovum, where the further fertilization takes place and the further development occur. So it is just the transfer of the ovum from a donor female to the healthy female, but unfortunately who is not able to produce the ovum from the ovary. So this technique is known as a gamete intrafallopian transfer. Now the third method is called as intracytoplasmic sperm injection. It is similar to the in vitro fertilization or test tube baby, but the problem is that these sperms are directly injected into the ovum because sperms do not have a viable acrosomes which cannot penetrate the ovum. So in such cases, in the laboratory conditions, the sperms are made to enter the ovum, followed by which the embryo develop, like the embryo transfer is there. That embryo transfer is done, which we have already studied in the first case, that is a in vitro fertilization. The third method is the fourth method is the artificial insemination. In this is used where the male partner are unable to inseminate. That is, are not able to shed their sperm into the vagina of the female. Maybe they have an impotency, that is, the penis is not functional, or they have a low sperm count during the ejaculation. In such cases, the sperms are collected from the husband and or the donor. If the husband is not so viable, the sperm may be collected from the other donor, sperm donor, and they are artificially introduced it into the vagina or the uterus of the female, where the further fertilization and the development takes place. So this method is called as artificial insemination, where the male partner is unable to inseminate. So these are the methods which can be used to assist those individuals which are infertile and make them fertile. Even after these methods, if unfortunately person is not able to conceive pregnancies, in such cases, the other method remains is the adoption of the children. So these are the methods which are used, and very commonly asked question, what are ARTs and what are the various methods used. One is a in vitro fertilization or embryo transfer, where it consists of two, zygote intrafallopian or the intrauterine transfer. The second is the gift, third is the ICSI, fourth is the artificial insemination.